Okay, our speaker tonight is Debbie Hill. Debbie is one of ours, and she graduated from here following her brother Matt. She graduated from here in 2009, and I think she was a spectacularly successful undergraduate. I think she was the top undergraduate of her year, or joint top undergraduate of her year, and she also won a prize which was for the best science, engineering, and technology project in the UK that year. So she has gone on from this place to learn about NMR and hyperpolarized nuclei at the Cancer Institute, and she's shortly off, I think, in a couple of weeks to her next position in Norway. But anyway, Debbie tonight is going to tell us about why it helps to run in a corridor.
So yeah, thank you for the lovely introduction. I'll just dim this slightly so you don't have to look quite so much at me and a little bit more at what's on the slides. So yeah, it's an absolute pleasure to be here and to talk to you. A little bit strange to be on this side of the lecture theatre, and yeah, and it's a real pleasure. So I'm hoping today to give you a little bit of a flavour as to how we can link physics with medicine and improve cancer diagnosis using MRI. So you've already had a little bit of a little bit of an introduction, but I'll take you back right to the start. So it started here for me about 25 years ago with this young man here, who's my brother Matt, sitting over there in the audience. Matt always loved physics, and he always loved learning about how things worked and how to break things, more importantly, and then how to put things back together, hopefully. And so with Matt, he always knew he wanted to do physics, and he was a really big influence for me as to why I kind of got into physics and how I found it interesting. But for me, things were a little bit more complicated than that, and I kind of enjoyed the biology and I liked the chemistry as well, but Matt said he wouldn't give me any credit unless I did a degree that had general relativity in it, so I had little choice. So I went to college and I studied chemistry and physics and also maths and biology. My physics teacher is here from college. It's great to see him here, and I had a really good time there. I actually found physics and maths to be the most challenging of my A-level subjects, and persevered with them, and I always found them very interesting, but still I did find it kind of a little bit daunting at times. But during my AS, between the summer of AS and A2, I had a Nuffield bursary here at Sussex where I worked with Ben Varco, and I was designing some microwave cavities for use in a quantum computing experiment. This gave me my first kind of taste of what research was like, and I really enjoyed doing that, and kind of was really, really excited then when Sussex invented this new degree, which was physics with a research placement. Now, anyone here that's kind of still looking to go to uni, if you're interested at all in doing research, then I say look up this research placement degree, because it's really, really, really good and gives you a good taste as to what doing research is like. So yeah, so I was really excited to come to Sussex, and I had the pleasure of meeting this young man who's also in the audience back there, Pete Hurley. So this is me and Pete in the third year MPhys lab, and Pete was a massive influence on me as well during my undergrad. He showed me what true persistence meant when he explained to me over and over and over again what my problem sheets were asking me to do. So I've got a lot to thank for that. And I really enjoyed my time here at Sussex, and I think that was a big part. So for the research placement, I spent a lot of the summers with Mike Hardiman in his lab, who are aiming to improve the sensitivity of the neutron, measuring the neutron's electric dipole moment. And in the lab, so I was working here with Katerina and Andrew, who's over there, and I told him I would put an embarrassing picture if he didn't show up, so it's good to see him here. So in the lab, I got my first taste of what magnetism was about and low temperature physics, and this was a really interesting subject for me to do. I also learned how to make the best cup of tea, which Mike is adamant that didn't happen, because he says he's never seen me make a cup of tea. but trust me, it was definitely one of the most important findings of my time. One of the highlights of the research placement was going to the ILL and seeing the main experiment in the flesh. So part of my, my project work was to make small scale model designs of this experiment and to test their properties. So having the chance to go to the main experiment was really nice, and it was an exciting thing to be able to see. So when I was coming to the end of my time here at Sussex, I had to make a choice of what to do next, and having really enjoyed all the research placement projects, I knew I wanted to get into research, and doing a PhD would be the most logical step for that. But one thing that I wanted was a little bit more kind of applied physics. I wanted to have something that affected people a bit more. So I started looking into medical physics PhDs, and I found this one at the Institute of Cancer Research. And the degree at Sussex doesn't have that much medical physics in it, so I was going into the interviews with not that much medical physics background, but a lot of research experience from the research placement. And that was one of the main things that set me aside in the interviews for the degree, for the PhD. And I think, again, not to ram home how good the research placement degree is too much, but I would say it's a really good thing to be doing if you have the option. So this brings me on to what I want to talk about today. So the main subject area is magnetic resonance imaging. And I want to tell you, first of all, how that's used in cancer diagnosis and in determining cancer treatment, but also how we're improving that now, and how my PhD has kind of helped to build on that. So those of you that are unfamiliar with an MRI scanner, it's a big magnet here, big donut-shaped magnet, and inside there is a load of liquid helium. So it's cold, it's superconducting, and it's really, really strong. So just how strong is this MRI magnet? So just to put it into perspective, we've got the Earth's magnetic field, about 50 microtesla. And we've got a scrapyard magnet here. So these things pick up cars and big chunks of metal, and they're around about 1.5 Tesla, which is 30,000 times stronger than the Earth's magnetic field. Whereas the clinical MRI magnets we use go all the way up to 7 Tesla, which is 140,000 times stronger than the Earth's field. So we've got some really, really strong magnets here. And the reason we need these strong magnets is to get these nice images. So I don't know if you want to play Guess Who, but these are some MRI images of myself. So we've got a brain, which is probably an important thing for me to point out to you. But one of the really nice things about MRI images is you can see this nice tissue contrast. So here you can see a bit of a kidney. I've got weird kidneys, so you can only see one of them, but there is another one in there, I promise. You've got a liver here. You can see the spine and the bones. So one of the reasons why MRI is so good at cancer diagnosis is you can see the difference between these tissues. And I'll show you a little more of the images later on. But first of all, how do we actually get this MRI signal? So MRI is essentially looking at where the water is in your body. So we're really lucky in that there's about 70% of our bodies are made of water. So that means we've got access to loads and loads of signal. And as I said a minute ago, the contrast is different in different tissues. So that's why MRI is really good for diagnosing problems or cancer. But in particular, we're actually interested in looking at the hydrogen when we're looking at MRI images, and not just going right into the atom now. So here we've got the hydrogen atom, and we're particularly interested in the proton, which is the nucleus of the hydrogen. Now, the nucleus has this property called spin. Now, I don't want to labour what spin really is, but I'm just going to call it spin for now, and it's a really important property in MRI. So we want to play with this spin. So if I were to take a box of water, which is here, I've got all these spins, and at room temperature, with no magnetic field, these spins are just randomly distributed around. They're just sort of doing what they like. But then if we were to turn on a magnetic field, like this, the spins will either align with the field or against the field. So we've got a magnetic field here. The spins are either aligned with or against it. Now, with this alignment, there is an energy associated with the spins. So the spins that are aligned against the magnetic field have a higher energy than the spins that are aligned with the magnetic field. Now, this is the crux of how MRI works. So if you want to get a signal, we're interested in the difference in the number of spins that are aligned with the field and the number of spins that are aligned against the field. So here we've got 7 on the bottom, 6 on the top. You find the difference between the two of them, and this is the kind of maths I enjoy. So you've got a difference of 1. So this would mean you've got a signal here because there's a difference in the number of spins up versus down. In reality, the maths is a little bit harder than that. I don't have this many fingers, but here I'll just introduce this term polarization. So the polarization is the difference in the number of spins up and down, what we just looked at. And in reality, we have actually 5 in every million difference. So this is for water protons at room temperature. So that means if I were to draw all of those spins out, I don't really want to draw a million, but so if you imagine this being a million spins on the top, we've got 1,000,005 spins on the bottom, so there's a difference of 5 in every million there. So this is actually quite a weak signal. Now, if you, this equation here is important, so the magnetization. Now, the magnetization is actually what is the signal strength, and you can see that if you increase the polarization, you increase your signal strength. But as I've just said, at room temperature for protons, it's very, very small number, so a very weak signal, and this is why MRI can take quite a long time to acquire the picture, to make your picture. But we're lucky because at room temperature, in the body, we've got so many protons. We actually have a big N here, so N is the number of spins, so the number of spins in your body of water. This is great. So we're actually able to get some good pictures to help us with cancer diagnosis. So that's enough of the first kind of physicsy part. I'll show you some pictures now if we can all have a look. So here you can see some pictures of people with cancer. So here's a brain and a very obvious brain tumour here. You've got some breast tumour here, and this one is a prostate. So the contrast is actually different here, but you can see the tumour here outlined with the arrows. So as you can see, MRI is really, really good at pointing out these cancers to us. So why is it that I'm here telling you that I want to improve the technique? You know, it's already pretty good. But the answer to that lies in the cases where you have something a bit more complicated going on. So have a look here. So this is a cross-section through the middle of a person. You can see here, he's not got weird kidneys, he's got 2. And you can see the spine here. So this big lump up here is a tumour, and that was before treatment. And this chap was given some therapy, and he went away. He came back for his post-treatment scan, and you see the tumour's got a lot bigger. Now, from the imaging alone, you say, this is bad news. We need to give him another treatment. We need to do something. Fortunately for this guy, he had another scan, which was called an FDG PET scan. So in FDG PET, you take a radio-labeled sugar, so it's a radioactive sugar, and that is injected into the body. Now, the cancer is really, really hungry, and it wants to take in this sugar. And in this image, black is bad. Black means lots of this radio-labeled sugar is stuck there. So before treatment, loads and loads and loads of sugar stuck in this tumour. But after treatment, it's gone. So in this case, this person has responded to treatment, but it hasn't shown up in the image just because of the type of drug we have used. And so it's really important to point out that we need other modalities to look at what's going on rather than just relying on imaging alone. So there are some limitations with this. So the FDG PET scan, because it's radioactive, you wouldn't want to do that in children, and you wouldn't want to do that very frequently in anyone, because you don't want to have exposure to that much radiation. So what I'm here to talk about today is how we can get information that's similar to that, but without being dangerous and without exposing anyone to anything hazardous. So we do that using a technique called magnetic resonance spectroscopy. So MRS works in exactly the same way that I described for MRI. We're still looking at the difference in population levels of the up compared to the down spins. But instead of looking at where the water is distributed, we choose a little box here, and we look to see what different chemicals are present. So that the height of the peaks on this graph represent the concentrations of different chemicals. And cancer looks very different to normal tissues, so you can characterize what kind of cancer it is and how aggressive it's likely to be, and look for different properties. But the problem with looking at proton signals in the water is that you have this huge water signal there that's fantastic for us for imaging, because that's exactly what we want to see there. But if you're interested in anything that's not water, then you're in trouble. You can see these tiny little blips down here, and it can be quite tricky to remove the water from your spectrum if you're interested, especially if you're interested in something quite close to it. It can be a nightmare. So our solution is to look for something different. And we're interested in this molecule, carbon-13. So there's tons of carbon in your body, but this is all predominantly, about 99% there, carbon-12. And carbon-12 is invisible in MRI. You can't see it. Whereas carbon-13 has this property of spin, because it's got this extra neutron. And so we can see it using MRI or MRS. And the important thing is that it's not radioactive, so it's not harmful. And the body will take in carbon-13 and use it in exactly the same way as it would use anything that contained carbon-12. So it's used identically, and that means we can monitor things like metabolism, which I'll get onto in a minute. But there's always a downside, and the downside here is that we have much smaller signal strength. It's only a quarter of the signal strength of proton, and there's not very much of it. But we can get around that by making carbon-13, so we synthesize it, and then we can inject it into the body and see where it goes. So cancer is really, really, really greedy, and it wants to eat this carbon-13 sugar. And that's great for us, because if we can inject in this carbon-13 sugar, we can watch what the cancer does with it. And any of you that have ever gone for a long run, the next day you come back and you're like, oh, my legs are really achy. That's because your body has undergone anaerobic respiration, and it's generated lactic acid. So cancers do that all the time. That's their kind of base level of metabolism. So they will take in sugar and they'll turn it into lactic acid. And normal tissues don't do that, and they don't do it that quickly. So cancers will stand out against normal tissues, and you can actually see this metabolism happening. But we need enough signal to do that. So we come back to this equation, the magnetization equation, where we know what our limiting factors are. So for carbon, we don't have very many spins. So we're not looking good so far. We know that this gyromagnetic ratio is small, so this is where the quarter of the signal strength comes from. And the polarization, that's also really small, because we're looking at a nucleus and there's not very big difference between the population of these spin states. So really that's it. We should probably just go home, and it was a nice idea, but unless we can cheat. And this is what we do. And we cheat by exploiting an electron. So electrons are fantastic. Here you can see the polarization against the temperature. So this is fairly, warmish, and this is really, really cold. And in our experiment, we're around about here, about 1 Kelvin. If you look at what the electrons are doing at 1 Kelvin, they're pretty much 100% polarized. Whereas you look at the nuclei, they're still, for the carbon, we're still down about 0.1%. So the electrons are doing pretty well at this point. And if you were to look at what their polarization was like on this spin diagram, it would be something like this. You've got a huge potential signal here. But our problem at the moment is this is the polarization of the electron, and we want it to be on the nucleus. We want to look at the carbon. So we need to steal this electron's polarization somehow and transfer it over to the carbon. And this is where dynamic nuclear polarization comes in. So this is the kind of crux of my thesis work using this technique. So the way that we do this is by taking a carbon-13 labeled sugar, and we put it into the sample cup, and we mix it with some free electrons. So this is where the stealing process begins, if you like. And we take that sample and we put it down to really cold. So I said we were operating about 1 Kelvin. So here is me inserting a sample into our machine. So it's called the HyperSense. And the HyperSense has a magnet here, which is about 3 Tesla. and it's also got a liquid helium bath. So when you put your sample in, you're cooling it down to around about 1 Kelvin, which is where we want to be to get the high polarizations. So another warning, I'm going to try and explain some of the physics again now. So I've had a couple of pictures, but a little more physics. So when you have your carbons and your electrons mixed together, you get a lattice something like this when you freeze it. So you have quite a lot of nuclei and a couple of electrons dotted around. And then when you put this into a magnetic field, you get this energy level diagram again. This is what we were looking at a minute ago. So this would be for the carbon-13 nucleus. And instead of saying the lower ones are aligned with the field and the higher ones are aligned against, I've called them alpha and beta. Now, the black letters here are for the carbon, and the energy gap here is separated by a space which is characteristic for the carbon. When we introduce electrons, we get some extra energy levels, and these energy levels all have red letters, and so you've got the electrons with a big gap between them, and the nuclei with a little gap between them. And so this is kind of how it looks when you draw in these characteristic gaps. So now if we put on the spin populations that we saw earlier from this graph, we can see that at this temperature, the nuclei have a very small difference in their population, but the electrons have a big difference in their population. And this is the way that this system wants to be. This is at equilibrium, it's happy. It doesn't want to change. And so, there we go. Big electron and small nuclear polarizations. So now what we do is we play around with this population distribution by applying a microwave irradiation. And we do that at this frequency here. And when you do that, you match the populations of these two states. So now this one has gone down a bit, this one's gone up a bit. And all the time you're applying this microwave irradiation, these spins won't do anything. They're tied up, they're swapping between each other, and they're not going to move anywhere. And all of a sudden, this electron up here is really unhappy, because in its partner state down here has just lost a load of spins. And this one is also unhappy, because its partner state up here has just gained a load of spins. And that isn't how it wants to be. That's not the equilibrium. And so, sneakily, we've tricked these electrons into relaxing down into this energy state from up here. So when that happens, you end up with you're satisfying the equilibrium between these two, but you've been sneaky and you've actually made a really big population difference between the nuclear energy levels, which is exactly what we want for increasing our NMR signal. And so this is what it looks like on the machine. You can see time along the bottom, and you can see your NMR signal up the top there. So over about an hour and a half, you can see your nuclear polarization building up just as we want. And this is fantastic. This is great. And just to show you how big an enhancement we actually get, here's an example of a spectrum that I acquired with urea, and it took 65 hours. When we do this DNP enhancement, 2 seconds and a much, much bigger signal. And this is the equivalent signal enhancement of taking a match and stretching it to the height of the Empire State Building. So we've got a really good signal enhancement here, and we can play around with this now. So that's the hard bit done. And I promise that's most of the physics done too. So what do we do? Once we've got the sample is cold at the moment, we've got this great polarization, but it's still cold, it's still frozen. And we want to dissolve this and make it into a state where you can inject it into a person and measure what happens. But it's tricky, because once you dissolve this, the signal starts dying away really, really quickly. So you have to act fast. And I'm sure from the title you can figure out now where the running in the corridor comes in. So the physics is done, and I've now got a reward for those who have managed to stay awake, which hopefully is a video of an experiment in action. So this is the screen of the HyperSense. You can see the solvent heating up there. That's the dissolution buffer heating up. When it gets to 10, that means it's action stations and everyone has to run like mad. So once it reaches to 10, the solvent will be shot down into the solid sample. It will dissolve it, and the liquid will come out here. So this is my supervisor Tom, who's trying to not look too nervous. And this is me. So I'm just speeding up a little here. You're going to see me take some of the sample out. Tom will check its pH, and then if I get the green light and it's all clear, then it's time to go. Right. So there you go. It's coming out. Give it a little shake. So I've drawn up enough. Tom's checking the pH. He says it's good. And then we go. And we have to go not too far. We do have to go. And then we're in. So that's the MRI magnet, and I'm going to do the injection. So if you were then to go to the control room and watch if you were going to go to the control room, hopefully, and watch what happened, you would see this. So you've got noise there, then bang, there's your pyruvate, this big peak here, so this is your sugar, and your cancer's making lactate. And you can see or lactic acid. So you can see it build up and then all die away. Now this is sped up, so this would ordinarily happen over the course of about a minute or 2 minutes, depending on which magnet we were using. But then you see it's all gone. So we have a short window of time where we actually get really, really nice data, but then it does all die away quite quickly. So if I was to plot that out in a time series, you would see something like this. So you can see where we injected in the sugar, and then you can see the lactate building up and dying away. So at this point, I've sort of already slipped into sciencey speak. If I say pyruvate, I mean sugar, and if I say lactate, I mean lactic acid. It's essentially the same thing. So from this, we need to do something which is quantifying the data. We need to make a way of saying, when we did this experiment, it had this much, and when we did this experiment, it had that much. And the way that we quantify it is by doing this kinetic modeling. Now this is the most important parameter here, which is this K1 or K+1, which tells us the apparent rate of pyruvate being turned into lactate. So when cancers are really aggressive, this will be really fast. And as cancers, if you give a treatment and it's worked, you would expect that rate to go down. Or for less aggressive cancers, you would expect it to be lower. And so that's what we measure. And we measure that by doing this thing called kinetic modeling. So this is something that I spent a good couple of years of my PhD in optimizing. And you can see here there's some quite complicated equations here that actually model the chemistry and the biology of the reaction. And you can see from the fits that they're pretty nice, and it works really well. And we're happy with how it was, with how it's worked, and it allows you to actually get a number out for this. So if we look at some data, here you've got some cancer cells that we've treated. You can see here the lactate curves before and after treatment. And you can see that there is a massive difference in these two curves. And then when you do the kinetic modeling and you figure out what these K values are, you can see a really, really big drop. So it's fairly obvious here that that it's worked and that the treatment has worked. And so this is what we're hoping to take onto the clinic and to say, this is how we should analyse the data, and this will give you an earlier marker of treatment response. And so I was very excited about this and put this work in for a conference in Australia, where I definitely didn't go on holiday at all and see kangaroos. So I was there happily showing off my data in Australia, and a chap comes along to my poster. So I'm explaining to him what we've done. And he seemed interested, and he seemed like, this was a really good idea. And then I got on to telling him about all this maths and about the kinetic modeling, and his eyes kind of glazed over, and I thought, oh dear, this guy's not a physicist, he doesn't know what I'm talking about. Actually, worse still, this guy was a clinician, and he was the person I needed to explain this to. If this was gonna work, the clinician needed to know what I was talking about. So he made it very clear he wasn't really interested in the maths, and he just kept looking at the graphs and saying, there's a change. And so he was staring at these graphs, and eventually we sort of said thanks very much and went away. But I was kind of disheartened a little, because I thought, if this is going to work in the clinic, I need to be able to do something that a clinician will understand, and it needs to be able to be analysed by a clinician. So I jumped back on the first plane back to the lab, and go over there, back to Sutton, where I sat at my desk for a while, and I thought, how are we going to do this? How are we going to make this easier? So I was weighing up the kinetic modelling against some other method. So what could we do that's easier? And it's clear to me now that because you've got this signal that's dying away, you're actually trapping an area under the graph in both cases. So what if you were to just measure how big the area was under the graph and see what the difference was before and after treatment? And so that's exactly what we did. And we looked at the maths of this, and so this is the correct way of writing what's the area under the graph for lactate and for pyruvate. And when you go through all the maths and you simplify all these things out, you end up with this really nice relationship between the thing that we're interested in the kinetic modelling and the thing that we can get by colouring in under the graph. And so I was thinking, great, maybe this is something the clinicians will like. So the way that we check if this works is by plotting I plotted all my experimental data. And you can see here you've got your K from the kinetic modelling, and you've got your lactate divided by pyruvate signals, and there's a really, really nice correlation between the two of them. And this was exciting for us. We thought, this means that you can bypass the kinetic modelling if you want to, and you can just plot the area ratios, and you get something that gives you the same sensitivity of detecting treatment response. And this was something I was hoping maybe I'd get a better result from a better response from the clinicians about. So I put this abstract in for another conference in Philadelphia. So I shot back over in the plane, and for those of you who know Philadelphia, it's the land of Rocky, and I was hoping I wouldn't come into too many confrontations while I was there, especially with clinicians, but I was ready, had the Eye of the Tiger going on my MP3 player. And so I went to this lecture, and I had an oral presentation, so I was looking out and thinking, right, there's got to be some clinicians out there, and I spotted one. And I put forward this area under the curve ratio, and I was very, very happy when his response was slightly different to the previous one, and we thought, great, we're getting somewhere, this is awesome. So we then wrote up the work, and I was really happy to have it published. So hopefully now around the place there are other clinicians having similar responses and other scientists having similar responses to this analysis method, which is a simpler method and should be really useful in the clinic. So that's kind of my major part in what we've done. But I want to tell you now about how the DNP field as a whole has improved in terms of clinical applications. And coming back to the kind of motivation for this was this FDG PET study. So we were after something that gave this kind of information, but without using radiation. And we've got these people to thank for this work. So this is John Kahanovitz, Dan Wieneron, and Sarah Nelson, and they're all professors in the UCSF Institute in San Francisco. And I was really lucky to have the chance to spend 6 months of my PhD there while they were doing the very first clinical trial of DNP. It was a really exciting place to be. And so I want to show you a couple of their results. So if you remember from earlier, I showed you this picture of the prostate cancer. Can anyone point out where the tumour is? Great. Where the red arrows are, right? Excellent. That's what I was hoping you'd say. So we use DNP to see, we put this grid over to get to acquire the spectrum. And this is what we saw from these points on the graph, on the image. And when you actually draw them as like a heat map, not only do we see a region here where we know the tumour is, we see another region over here, which is really, really interesting. If you can compare that, I mean, I don't know, I've looked at this image quite a lot, and I can't see anything here. I know it's not the highest quality, but I can't see any evidence of a tumour here. Whereas the DNP says something really different. And this was really exciting for the DNP field. And this person actually had a biopsy taken of the tissue here and the tissue here. Now, biopsy is where you kind of remove a little bit of the tissue, it gets sent off to the lab, and they analyse it to see whether it's normal or cancerous. And it was proven that this area was actually cancerous as well. So DNP's not only found this bit, but it's also shown that this guy had another area that we needed to be aware of. And so this is exciting progress in the field. And so because of how good this research has been, there's been a lot of developments in the way of DNP's clinical applications. And they've got this now dedicated spin lab, which looks slightly different to the HyperSense I showed you earlier, and also means that people have less far to run because you can actually put them right next to the scanners, which is great. So overall, I'm going to sort of wrap up there and say that hopefully you've seen where my work sort of fitted into the overall field of DNP. And if there's anything important that I'd like you to take away is if you see a physicist running in a corridor, maybe you'll first of all step aside, let them run past, maybe cheer them on, unless they look like this, in which case you should run after them and try and keep up, I think. So with that, I'll say thank you very much to, first of all, to my supervisors, who are these three, Tom, Ewan, Lee, and Martin, and then people that have been directly related to the work, especially Neil for helping me out a lot with this presentation as well. But it's not just the people that you work with, it's all of the other people that make a big difference to your experiences as well and keep you going. So thank you to all of them as well. And also thank you all for turning up and for listening to me for the last 40 minutes. So I'll stop there and happily take any questions.
Okay, thanks, Debbie.
Right. Do you want the lights?
Yeah, I think with the lights. Okay, do we have any questions? Maybe I could ask you one to start off with. You're talking about injecting, you've got a free radical in there, which is carrying the electrons.
Yeah.
I was always taught that free radicals were things
Bad.
you shouldn't have because they gave you cancer.
Absolutely. So in the...
Unless you eat lots of broccoli.
Yeah, we don't force feed patients broccoli before. So in the clinical experiments, the free radical is completely removed from, so it goes through a column and is removed from the solution before it's injected.
Oh, I see, okay.
And there's actually some really neat advancements in that. You, instead of, this is something that was in a conference recently, instead of having, like mixing the radical in to the liquid, they have the radical trapped in like a silicon matrix, which is really absorbent. So you can squeeze your sugar onto this matrix, the polarization happens, and then all you need to do is just wash it out, and the radical's actually trapped in that matrix, which I think is a nicer way of doing it.
Can I ask, you can tell an API?
You can, in DNP, in my project I've done mostly carbon. We've done some nitrogen. Nitrogen's really good. It has a really, really long T1, so that the signal lasts a lot longer. But the problem with compounds that contain nitrogen is they're usually involving quite slow transporters, so we didn't actually see anything when we injected it into cells and things. But people are also looking at silicon a lot. That's something that silicon has a huge signal, a really, really long signal, so that's another thing that's been pretty good.
There's a belief that helium-3 was used to some extent.
Yeah.
Polarized helium-3.
Yes.
And I think it was stopped.
Well, so helium-3 is used in, so there's a hyperpolarized gas, and is used a lot in kind of lung function studies. And yeah, it's not particularly done using DNP anymore. It's done using things like optical pumping and things like that they use. But I think helium shortage is sort of hitting everyone a bit. They have swapped over to xenon. They're using xenon and things like that. And maybe, yeah.
I have a question. Do you know your clinical polarizer, right? So that's at clinical trials. You're still using the carbon 13, is it?
Yes.
And the signal strength is still 1 minute.
So you have about 3 minutes of signal. The signal dies away. So it's called the T1, which characterizes this exponential decay of your signal. That's about 1 minute. So that means that you have about 3 minutes or so to actually acquire the data. So
you can only get data over there.
yeah,
And that's it. So
yeah. So that's why it's so important to do things so quickly. But with the spin lab, it's better. Like they have a better system of removing the radical and getting it from the solid into the liquid where you can inject it is a lot quicker now. So the results that I was showing there is actually on the very first system that was made, and it took about 40 seconds to get from the dissolution to injection, and they've reduced that down now, I think, to about 20. So it's getting better.
So that means from this data that you receive, can you actually determine right then that there is a tumor, there isn't a tumor, and so on and so forth? So you can give patients results.
You could give them,
faster as well?
yeah. I mean, you wouldn't give it to them while they were lying on the bed, but you essentially you would need to, like, the practical side of it would be get the data off and analyze it and give it back. We're talking, so changes in tumour volume can take weeks to occur, and changes in metabolism can happen within 24 hours or less. So you're talking about finding response to treatment a lot earlier than looking for a change in volume. And I showed an example where change in volume wasn't an appropriate way of determining treatment response as well. So it's good in that regard too.
On one of your scans, the second one where you found the tumour which wasn't shown on, it seemed like quite a specific region.
Yeah.
Are you thinking like trying to broaden out if you don't know where the tumour
Yeah, that's a really good point. So in the example I showed, that was the way they had set up the acquisition, but you can acquire data over the whole region. I think what's more likely to happen is, in the first instance anyways, people often have a diagnosis beforehand, and you know where the tumour is likely to be. In this case, I think we were they were lucky, but this person would have found that tumour using something else, I think. But yeah, you can put a grid over the whole area. It's just a case of playing with the signal and trying not to lose too much of it within that time.
Another question: how fast is the scan? I mean, if you've only got a couple of minutes to do if you've only got a couple of minutes of lifetime of the hyperpolarized species, how long does it take to do the MRI scan of the region?
So when you say, for example, the example I showed was the most simple sequence we could use, which was the pulse acquire. So we weren't localizing; we were just acquiring over the whole region. And there, the spectroscopy sequences are really quick. So I was repeating the scan every 2 seconds. So
but the image.
When you do an image,
yeah, the image takes quite a lot longer. Doesn't it?
Not when it's hyperpolarized. You can get away with you do tricks. So you use kind of ultra fast TE and sequences, and you use low flip angles, so you don't kill the signal in your first acquisition. But a lot of the time it's kind of an EPI, so you play with gradients rather than playing with RF pulses.
What inspired you to want to do this kind of research? Like, what motivated you to do it?
So I think that the first thing was just wanting to. So I love physics, but I found that working on physics that was kind of more pure physics didn't feel like I was getting enough of the application into people, and I've always been interested in the chemistry and the biology. So I kind of felt like I wanted to learn what physics was like when you applied it to a more biological system. So that was kind of how I found the Institute of Cancer Research. But I think actually getting this project was, I don't want to say it was luck, but I actually went along to the interviews for a different PhD, and this project was advertised on the day that I was there, and it was just like, oh, by the way, we've managed to get another project in. So I kind of didn't have much time to think about it. So it was kind of just like, oh yeah, that sounds cool, and I went with it from there, and it's been a really rewarding area to be in. And I wish I'd have known about it before, because I think if I'd have had longer to prepare for the interview, it might have been a bit less scary. But yeah, it was a nice question. Hi, Van.
Just where it's going to take you next, everybody, and what your hopes are for your next
Yeah.
work.
So my next, so I'm starting a postdoc in Norway, in Trondheim, on the 1st of November. So I'm leaving in not very long. And my next project's actually going to be looking at prostate cancer. So I'm honing in on a specific type of cancer. But actually the group that I'm joining don't have DNP facilities, but they're interested in getting them. So I think I'm kind of going to be bit of a sounding board for them. But I wanted to broaden my horizons a little bit, because I feel that I've learned DNP now, but I don't feel like I have that much strength in the kind of normal imaging side, so the kind of using other techniques to find out similar information. So I'm hoping to gain some of those experience or some of that expertise over the next sort of 2 years or so, and see what happens next.
Questions? Okay, the one question I was going to ask you, when you were injecting, you were shown as running and injecting into something. I presume that was a dead dog or a rabbit or something. It wasn't actually a person.
That wasn't a person.
Good.
No.
Just check on that.
Yeah. Just for the sake of the recording, it definitely wasn't a person.
Right, we don't have to talk to the RSPCA about
No.
this either. Okay, well, I think I'd like to say thank you to Debbie, and to wish her well in her next venture, and also to point out that although she's doing applied physics with using magnetic resonance, in fact she learned some magnetic resonance here doing some
Absolutely.
really blue sky stuff.